Hello, my name is Ominda, and I'll continue with the lecture on histology of the respiratory system. So, how the conducting portion of the air, it bifurcates a sternal angle of V to form primary bronchi. These two will divide to form secondary bronchi, which are the lobar bronchi. Okay, the lobar bronchi. Then there's one secondary bronchus on each lobe of the lung. So the right lung has 3 lobes, the left has 2. So there is one secondary bronchus for each lobe of the lung. So there are 2 lobes of the left and 3 lobes of the right. And these secondary bronchus, just like primary bronchus, they have the same anatomy as the trachea. Okay. Then the secondary bronchi, what happens? They now branch to tertiary bronchi. So these tertiary bronchi, they eventually continue to branch, and they get smaller. So from primary, secondary, tertiary, as they keep branching, the caliber, the diameter becomes smaller, number one. Number two, there's loss of cartilage. Remember, there's lots of hyaline cartilage in the tracheal ring. So as you continue dividing to primary, secondary, tertiary, you lose the cartilage. It becomes less. Then when you lose the cartilage, what happens? Eventually, the airway is now called bronchioles. So the bronchi, primary, secondary, tertiary, they have cartilage. When you get to bronchioles, you lose the cartilage. So the tertiary bronchiole, supply a lung segment. Tertiary bronchiole. Secondary was supplying a lobe of a lung. Tertiary supplies a lung segment. Primary bronchi supplies each lung, right and left. There are 2 primary bronchi. Secondary bronchi supply the lobes of the lung. So there are 3 on the right and 2 on the left. Tertiary bronchi supply the segments. So one tertiary bronchus, okay, is distributed to the bronchopulmonary segment. The tertiary bronchi give off terminal bronchioles, which give rise to respiratory bronchioles. So tertiary bronchus to terminal bronchioles to respiratory bronchioles. Tertiary bronchiole to terminal bronchiole to respiratory bronchiole. So one respiratory bronchiole, okay, therefore gives an alveolar duct, which has an atrium, and then from there you have alveolar sacs, then the alveolar cells, which are closely associated with the blood vessels and neurovascular structures, lymphatics and nerves, to form the functional unit of the lung, which is the blood-air barrier. Okay, so trachea, nasal cavity to pharynx, larynx, trachea, primary bronchus on each lung, right and left, secondary bronchus on each lobe, 3 on the right, 2 on the left, then tertiary bronchus to each bronchopulmonary segment. Then from tertiary to terminal bronchioles, then respiratory bronchioles. From respiratory bronchioles to alveolar ducts with an atrium, then alveolar sac, alveolar cells. And these alveolar cells are in close proximity with capillaries and nerves and lymphatics. And at that portion, we form the functional unit of the lung. So the alveolar is a functional unit of the lung. So together with the associated capillaries, you form the blood gas barrier. Okay, so the bronchioles terminate in alveolar. The respiratory bronchioles will eventually terminate in alveolar via alveolar ducts, and the walls of the alveolar are 1 cell thick, so it's simple squamous. Okay, that's why we say the lungs are lined by simple squamous epithelium. It's because the alveoli are 1 cell thick, single layer of flattened cells, and they're covered by capillaries. They're in close proximity to capillaries. So alveolar are the smallest structural and functional unit of the respiratory system. So alveolar are the functional respiratory units of the lungs. So the respiratory bronchioles, remember from terminal bronchi to respiratory bronchioles, they're lined by simple cuboidal epithelium, okay, and it could be ciliated. There's a presence of Clara cells. These Clara cells produce some surfactant, and we have alveolar opening. So alveolar now opens, okay, from these respiratory bronchioles via alveolar ducts. So we have 2 types of alveolar, type 1 and type 2. Type 1 are the simple squamous, single layer flattened cells for gaseous exchange. So you're able to have a loud diffusion. Okay, so this covers 95% of the alveolar surface. Then type 2 are cuboidal. Type 1 is simple squamous. Type 2, they're cuboidal. Their main function is to produce surfactant. Type 1, the main function was for gaseous exchange. Type 2 produce surfactant. So these are approximately 60% of all pneumocytes. Then we have alveolar macrophages. Okay, we have alveolar macrophages. So the alveolar, we have type 1 pneumocyte, type 2 pneumocyte, and alveolar macrophages. These are dust cells. Macrophages, so they are members of the monocytic phagocytic system. Okay, so they have an immune role. What is surfactant? It's a lipoprotein, surface active lipoprotein, and it contains phospholipoprotein. Okay, the surfactant is produced by type 2 pneumocytes, and the surfactant is a lipoprotein. That means it's made up of lipid and protein. So what does that mean? The portion with the lipid is hydrophobic. It does not, it's hydrophobic means non-polarized, like does not dissolve with water. The protein portion is hydrophilic. It can bond with water. So by adsorbing to the air-water interface, remember there's an air-water interface. What do we mean air-water interface? The alveolar contain air, and they're close to lungs that contain blood. So by adsorbing to the air-water interface, the interface between alveolar and the capillaries, yeah, the alveoli, the interface, the alveolar hydrophilic head groups. Remember, the surfactant has hydrophilic and hydrophobic points. So the head groups of the hydrophilic head groups in the water and hydrophobic tails that are facing towards the air. Okay, that is how they are going to interact with this surfactant. So surfactant has lipid, and the main lipid is the dipalmitoyl phosphatidylcholine. The lipid in the surfactant is dipalmitoyl phosphatidylcholine, and this usually reduces surface tension. So surfactant has both lipid and protein with both hydrophilic and hydrophobic ends, and even the air-water interface has both hydrophilic head and hydrophobic ends. So this surfactant contains lipids, dipalmitoyl phosphatidylcholine, that helps to reduce surface tension. So the surfactants help to increase pulmonary compliance, and help to reduce, or rather prevent, atelectasis. Atelectasis is collapse of the lung. So the presence of surfactant help you to prevent the collapse of the lung when expiration ends. You know, after expiration, you've removed most of the air from the lung. So you don't want the lungs to collapse, to undergo atelectasis. That is why the surfactant is there to increase the compliance of the lung. So it's able to facilitate recruitment of collapsed airways and regulate the alveolar size. That way, it also prevents fluid accumulation and has a role in innate immunity. But the main role of surfactant is to prevent atelectasis, which is collapse of the lung after expiration, by maintaining pulmonary compliance and recruiting the collapsed airways. So it also regulates the alveolar size. This is the histology of the lungs. So you can appreciate the alveolar simple squamous. So these are pneumocyte type 1, but adjacent you have cuboidal pneumocyte type 2 cells. And this pneumocyte type 1, where there's gaseous exchange, are in close proximity to the capillaries. What is blood-air barrier? So the alveolar lumen, the blood-air barrier in the alveolar lumen, there's a surfactant that is present. And then how do you describe this blood-air barrier? The main histological components are the type 1 pneumocytes. They are laying on a basal lamina. Then we have a capillary that is nearby, lined by capillary endothelial cells, also lying on a basal lamina. So those four are the components of blood-air barrier. Type 1 pneumocytes lying on the basal lamina, and capillary endothelial cells lying on the basal lamina. So the basal lamina of these two cells are fused, and that forms blood-air barrier. So these capillaries in the blood-air barrier are actually non-fenestrated. They are continuous. The endothelium is continuous. Okay, they don't have perforations, so they're non-fenestrated capillaries. But and then in the areas where the basal lamina of the two basal lamina are not fused, usually reticular and elastic fibers are present. Okay, so what is the function of blood-air barrier? Because it's between the type 1 pneumocytes and the capillary endothelial cells. So the alveolar pneumocyte is containing air. Capillaries are containing blood. You don't want air to enter blood because you have thrombus, or I mean emboli. Air is not supposed to be in blood vessels. So that's the function of blood-air barrier, to prevent air in gaseous form from mixing with the blood. It will not enter the capillaries. You just have diffusion, okay, of oxygen and carbon dioxide without having this in liquid form from blood. Then by the time they get to the alveoli, it's in gaseous form. So you don't want this air in gaseous form to enter the blood. So this is how gaseous exchange occurs. So you have your alveolar here with simple squamous, then you have capillary endothelium with simple squamous, their basal membrane are fused, and then you have carbon dioxide from blood to the alveolar and oxygen from the alveolar um, being reabsorbed. So there's a large surface. The adaptations are three adaptations of blood gas barrier, or sorry, the gaseous exchange surface has three adaptations. It has a large surface area for diffusion. It's very thin with simple squamous epithelium, so short diffusion path, and there's a concentration gradient to allow diffusion. So the carbon dioxide in the capillary is higher than in the alveolar, so carbon dioxide goes in that direction, and the oxygen is more in the alveolar than in the capillary, so it will be reabsorbed into the capillary. So those are the three adaptations of a gaseous exchange surface: large surface area, the short diffusion path, and a concentration gradient. So what are the cells in the respiratory epithelium? You have goblet cells that produce mucus, okay, to collect particulate matter and absorb noxious gases. Then you have number 2, ciliated columnar cells. This form the lining of the respiratory system. So they're ciliated, they're able to propel whatever, cilia will propel whatever inhaled debris, okay? Because when you inhale the debris, the mucus will trap it, then you need to get rid of it. So it's a cilia that will propel it upwards so that you can cough it and swallow it. Then we have columnar brush cells. Brush microvilli. Brush, when you see brush, it means we have microvilli. So columnar brush cells, there's numerous microvilli on the surface, and they have dendritic synapses on the basal surface. Dendritic synapses, so some nerve impulses are being transmitted. So what's the main function? You can guess: sensory. Okay. Then you have basal cells, which are short cells. Basal cells in any part of most part of the body, they are usually for regeneration. Okay, they are cells on the basal lamina. They do not extend to the surface epithelium. They are not on the surface of the epithelium. They are towards the basal lamina. So they help to regenerate the epithelium. Number 5, you have granule cells. Okay, these granule cells, they have granules in their cytoplasms, and they are cells of the amine protein uptake and decarboxylation. So they are neuroendocrine cells that secrete hormones to regulate the tract, the respiratory tract. So amine protein uptake and decarboxylation cells, which are neuroendocrine cells. We have Clara cells. These are secretory. They secrete surfactant. Surfactant helps to reduce surface tension of fluid and moisten the surface of the lung. Then we have alveolar type 1 forming gaseous exchange surface, and alveolar type 2, which produce surfactant. Okay, so we have metaplasia, where we talked of change of epithelium from pseudostratified columnar to squamous epithelium in the trachea due to irritation. Cartagena syndrome is genetic condition where cilia does not develop well, so you can't propel the mucus that has trapped dust. So patient will have recurrent respiratory tract infection. Then you can have malignancies of the lining of the bronchi, or malignancies, cancer of the lung, or bronchial carcinoma, and so on and so forth. Thank you very much.